Good day. So for this week's lesson, we will be tackling culture media preparation. So after the completion of this lesson, you should be able to define the basic components found in a culture medium, classify and differentiate the kinds of culture media used in a laboratory, and discuss and describe the procedure on how to prepare, sterilize, and dispense plated broth, butt or deep slant and butt slant culture medias. Okay? So for this day, we will only be focusing sa theoretical. So once we go back sa face-to-face classes, tsaka natin ipe-perform yung culture media preparation. So a nutrient material prepared for the growth of microorganisms in a laboratory is called a culture medium. Some bacteria can grow well on just about any culture medium, others require special media naman. That's why we have different types and classifications ng culture media. So microbes that are introduced into a culture medium to initiate growth are called an inoculum. So the microbes that grow and multiply in a culture medium are referred naman as the culture. So in order for us to have a culture medium, there are certain standards that we should meet. Okay, so what are those? Number one, it should contain appropriate nutrients. Okay? So certain organisms require certain nutrients for them to grow. So one of our examples of those are carbon source, energy source, nitrogen, minerals, vitamins, growth factors, and water. Okay? And then other certain bacteria, meron pa silang mas specific na for them lang. Okay? Yun. Next, it should have sufficient moisture. Okay? So sa moisture, it would also depend sa bacteria na gusto nating i-culture. Okay? So it should contain sufficient moisture, also properly adjusted pH, suitable level of oxygen, or none at all. Okay? So for the oxygen, it could be some bacteria require oxygen for them to grow, some naman does not. Okay? So oxygen for them are toxic. So it would depend. Okay, most bacteria grow best at a neutral pH value of around 7. Okay, fungi, however, prefer pH values around 5 for best growth. So, most commercial medias natin, yung mga nabibili natin, does not require adjusting the pH. Okay? So, ang ginagawa natin is, dinatagdan lang natin ng water. Kasi, yung mga prepared medias natin, specific na talaga siya for a certain organism. For example, SSA, EMB, meron na talaga siyang organism na para sa kanya lang. Okay? Okay. So, next is sterile. So, the medium should be initially sterile. Okay? Para when we culture, okay, once we added an inoculum, kung ano lang yung gusto nating i-culture, yun lang yung makikita natin. Kasi if hindi siya sterile, umpisa pa lang, so parang nawala na siya ng purpose. So, hindi rin natin makukuha yung mismong bacteria na gusto nating ma-isolate. Okay? So, we sterilized our medias using the autoclave then. So, 1 to 1 degree Celsius for at least 15 minutes then. And then, it requires incubation at a proper temperature. So, no matter how good you have prepared the culture medium, sterile nga siya, merong appropriate moisture, pH, and whatever. Kung hindi naman siya incubate ng ayos, hindi rin tutubo si bacteria. Okay? So, it requires incubation at a proper temperature. So, what are the common ingredients na makikita natin sa mga culture media? So, among this list, hindi lahat ng culture medium meron sa kanila. Okay? Yung may mga specific lang. Okay, let's go on. Number one, first water. Okay, for water, di ba yung mga nabibili natin culture medium prepared is in powder form. Okay, so ang nilalagyan na lang natin siya or niluluto sa tubig, parang gelatin. So the water that we should use should be deionized or distilled water. Okay, so hindi tayo pwedeng gumamit ng tap water kasi hindi yun sterile. Okay, number one, hindi sterile si tap water. And second, it can contain certain minerals, for example, calcium, phosphorus, magnesium, yon, that could react with the other minerals or ingredients in our culture media. Okay? So, next is the agar. So, agar, yung responsible kaya nabubuo yung media natin. Para siyang nagsosolidify. Okay? So, agar or agar-agar is prepared from a variety of seaweeds. Okay? Yeah. So, at certain concentrations, or at the concentrations normally used, most bacteriologic agars melt at about 95 degrees Celsius and solidify only when cooled to about 42 degrees Celsius. So, concentration of 1 to 2% usually yields a suitable gel. So, agar has some very important properties that make it valuable to microbiology. Okay? And no other satisfactory substitute has ever been found. Okay? Few microbes can degrade agar, so it remains solid. Also, agar liquefies at about 100 degrees Celsius, or the boiling point of water, and at sea level remains liquid until the temperature drops to about 40 degrees Celsius. Next is peptone. Okay, peptone is a complex mixture of partially digested proteins. Okay. So from number 3 up to the next slides, yun na yung mga specific. Hindi lahat ng culture medium merong ganong component. Okay? Number 4, yeast extract. So this contains a wide range of amino acids, growth factors, and inorganic salts. 5, malt extract. Consists mainly of maltose, starch, dextrins, and glucose. Contains about 5% of proteins and protein breakdown products and a wide range of mineral salts and growth factors. Number 6 is blood and serum. So these are used for enriching culture medias. Okay. So let's proceed with the classification of medias. Okay, so sinabi ko kanina, may iba-iba tayong types and classifications ng media. Okay? So we can classify them as two different types. So first one, based on phase of growth media. So we have liquid, solid, and semi-solid media. So liquid, as the name implies, liquid siya. Okay? Or yung mga broth. Okay? Solid, yung mga nakikita natin sa plate, sa tubes. And then, semi-solid naman is mas malapot siya kaysa sa liquid, pero hindi pa rin siya nagsosolidify. Okay? Next is based on nutritional factors. Okay? So, we have a simple media. Okay? So, simple media, usually, it's not specific. Maraming klase ng bacteria ang kayang tumubo sa kanya. Okay? And then, we have complex media and synthetic or defined media. And then, next we have special media. So, we have different types. We have enrichment, selective, indicator or differential, transport, and sugar media. So, iisa-isahin natin yan. And then last, we have based on phases of growth. Okay, so naulit na lang pala yun. Yeah. Okay. Yeah. So, liquid or broth media, okay, as I've said before, so, liquid talaga siya. Okay, so nutrients are dissolved in water, and bacterial growth is indicated by a change in broth's appearance from clear to turbid. Solid naman, yung mga nakikita natin sa sa plates. Okay, so made by adding a solidifying agent to the nutrients and water. So, yun yung agar. So, agarose is the most common solidifying agent. So the petri dish containing the agar is referred to as agar. Then. Next is semi-solid media. So for special purposes siya ginagamit. Agar is added to media in concentrations that are too low for them to solidify. Semi-solid media contains about approximately 0.5 to 1% ng agar, usually used then for motility studies. Solid agar contains 2 to 3% of agar. Okay. Next is classification of media based on nutritional factors. So this is also the same with the classification of media according to composition. So number one, we have simple media. Okay. Pag simple, so it only contains the basic nutrients required for the growth of ordinary organisms. So general purpose lang siya. For example, nutrient broth, nutrient agar. Next is complex media. Okay. So, complex media contains some ingredients of unknown chemical composition. Okay? So, unknown siya kasi hindi natin alam gano'ng karami bang, like for certain ingredient, kung gano'ng karami siya sa media na yon. Okay? So, kung gano'ng karami yung amino acids, yung nucleotide bases, vitamins, and yung other growth factors. So, hindi natin alam kung ilang percent ba siya out of the media. Okay? So those medias are called complex medias. Okay? So another example ng one of the examples ng complex media also is nutrient agar. Okay? So nutrient agar is also a uh, complex media. Okay? So hindi natin alam exactly kung ano ba yung composition niya. Okay. So, compared with synthetic or defined, okay, so sa defined, alam natin exactly ano ba yung chemical composition niya. So, yung individual components, they are weighed out exactly to make up the medium. Okay? Yun yung difference ni complex and ni synthetic. Okay? So, isang nabanggit natin kanina is the nutrient broth. So, nutrient broth is a commonly used complex medium, okay, in liquid form. Okay, kaya siya broth. Okay? So, it is a complex but also a simple medium. Okay? Kasi ginagamit natin siya to support growth of many organisms. Okay? So, hindi siya specific. Okay? So, it contains basic nutrients then na hindi natin alam yung composition. Okay. So, bakit natin nasabi hindi natin alam? Let's see different types ng nutrient broth. Okay. So, we have number one, meat infusion broth, meat extract broth, and we have digest broth. Okay. So, for example, meat extract. So, nagaling siya sa meat extract. Hindi mo naman alam yung certain compositions na meron sa meat kasi hindi naman ikaw yung gumawa ng meat, if that makes sense. Unlike with the defined, kunyari lalagyan mo siya ng 10% glucose, 10% sucrose, or whatever, alam mo. Okay? Or winay mo. Okay? Yun yung mga chemically or synthetic medias. Okay? So, this is, again, complex. Nutrient broth. Nutrient agar. So, nutrient agar is prepared by adding agar at a concentration of 2% to the nutrient broth. Okay? So, si broth, wala siyang agar. Okay? Pag nilagyan natin siya ng around 2% ng agar, magiging nutrient agar na siya. Okay? This is the simplest and most common medium used routinely in microbiology laboratories to grow non-fastidious bacteria. So, commonly referred to as the agar medium. Non-fastidious meaning, hindi siya maarte. Okay? When we say fastidious bacteria, yun yung mga mahirap i-isolate. So, they have a certain ingredient or certain nutrient na kailangan na meron sa agar para bago sila mabuhay. Okay? So, pag sinabing non-fastidious, so, yun, wala siyang specific nutrient na kailangan or demand. Okay? So, kaya niyang mabuhay on almost all of the agars. So, we have different types of nutrient agar. Number 1, semi-solid. Okay, so concentration of agar is around 0.5 to 1%. So, enables motile organism to spread but not non-motile bacteria. Next is firm agar. Concentration ng agar niya is 2 to 3%. So, next classification of media is the special media. So, this is also the same classification according to function or purpose. Okay. Number 1 is enriched media. Enriched is prepared to meet the nutritional requirements of a bacteria by adding the addition of substances such as blood, serum, or egg to a basal medium. A basal yung simple medium. Okay. So, dinagdagan na natin siya ng ingredient para ma-meet yung certain requirements ng bacteria na gusto natin i-isolate. So, that is enriched media. Number 2 is enrichment media. Okay? Enrichment naman is a substance, when a substance is added to a liquid medium which inhibits the growth of unwanted bacteria and favors the growth of wanted bacteria. So, sa enrichment, naglagay tayo ng ingredient that inhibits the growth of unwanted bacteria and favors the growth of wanted bacteria. So, it is a certain ingredient na ayaw ng ibang bacteria and gusto naman ng iba. Okay? Yun. And, take note, liquid medium siya. Okay? So, added to a liquid medium. Ang term natin doon is enrichment media. Okay? Number 3 is selective. Okay? So, selective naman siya when a substance is added to a solid medium which inhibits the growth of unwanted bacteria but favors the growth of wanted bacteria. Okay? So, kung si enrichment sa liquid, si number 3, si selective sa solid. Okay? So, actually, same purpose lang naman sila, na iba lang yung term kasi iba yung type ng media natin. Iba yung consistency ng media. So, pag liquid, enrichment yung magiging term natin. Kapag selective naman, solid yung magiging medium natin. Okay, so selective media is used to isolate particular bacteria from specimens where mixed bacterial flora is expected. So let's look at an example of a enriched media. So blood agar and chocolate agar. So many medically important bacteria are fastidious. So they require a medium that is even richer than nutrient agar. Okay? So one example of that is the blood agar. So blood agar ginagamit natin for isolation of streptococci, pneumococci, and Haemophilus. Okay? And then, medium, used to culture even more fastidious bacteria, is the chocolate agar naman. Okay? So, chocolate agar is also contains blood. Okay? Ang difference lang niya, ang blood ng chocolate agar is already hemolyzed. Okay? So, hemolyzed na siya. Also, another name ni chocolate agar is heated blood agar. Okay? So, kaya siya mas dark. Okay? So, yung agar kasi, yung blood kasi is naluto, kumbaga. Kaya mas dark siya in color. Okay? So, this one is used naman for isolation of Neisseria and Haemophilus species. Okay, another example ng enriched media, we have Loeffler's serum. Okay. So, serum naman ang inad natin sa medium. Okay. So, for isolation of Corynebacterium diphtheriae. Examples of enrichment media naman. So, again, enrichment. Enrichment is kapag liquid ang media natin. Okay? Liquid medium. So first, this is a tetrathionate broth. Okay? So tetrathionate inhibits coliforms while allowing typhoid or paratyphoid bacilli to grow freely in fecal sample. Okay? So fecal, as we know, our gut contains a lot of normal flora. So halo-halo sila dyan. Halo-halo yung bacteria. So when we want to isolate a certain type, so we use enrichment. Okay? Yan. So para, kunyari gusto nating i-isolate si typhoid or paratyphoid bacilli, so we can use this. Okay? This one is selenate broth. Selenate is used for dysentery bacilli. Dysentery bacilli. Alkaline peptone water naman, used for Vibrio cholerae from feces. Okay? Alkaline peptone water. Examples naman ng selective media. So again, selective ang term natin kapag solid medium siya. First is Lowenstein-Jensen. Ito yun. So this medium is used for Mycobacterium tuberculosis. Number 2 is deoxycholate citrate agar, DCA. Ito. Addition of deoxycholate acts as a selective agent for dysentery bacilli. So, isolation natin ng mga Shigella. Ng Shigella naman. Next is TCBS. Okay. So, this contains bile salts. So, bile salt is a selective agent. It makes sure the growth of only Vibrio cholerae and inhibits the growth of other intestinal organisms. Okay. So, next, indicator media. So, yung mga media na to contains an indicator which changes color when a bacterium grows in them. Okay? And lastly, differential medium. Okay, so, differential naman is a medium which has substances incorporated in it, enabling it to bring out differing characteristics of bacteria and thus helping to distinguish between them. Okay. For example, this one. Uh, Wilson and Blair medium. Okay. So Wilson and Blair has uh, incorporated, sorry, sulfite. So it has sulfite, bismuth sulfite. Okay. So si S-typhi, this is used for S-typhi, okay, reduces sulfite to sulfide in the presence of glucose. So, and the colonies of S-typhi have a black metallic sheen. Okay? So, pag nakakita ka ng black metallic sheen, alam mo na S-typhi yung inyong na-isolate. Okay? So, ayan. B, Klebsiella pneumoniae. So, pag Klebsiella pneumoniae naman ang na-isolate, ito yung magiging itsura niya, and then Pseudomonas aeruginosa. Okay? So, indicator, it has an indicator, nasa name niya na, to tell you what certain bacteria have you isolated. Next is McConkey. McConkey also is one of the most commonly used sa laboratory. Okay, so McConkey agar indicates lactose fermenting property. Okay, so pag lactose fermenter yung inyong na-isolate, it produces pink colonies, and non-lactose fermenter naman produces colorless colonies. Okay? Kasi meron siyang indicator, which is the neutral red reagent. Okay? So, lactose fermentor produces pink colonies. Si non-lactose naman produces colorless because of its indicator, which is neutral red. Ayan, example ng differential. So, sorry. Macaulay agar is a indicator media and also a differential media at the same time. Okay, so it differentiates non-lactose from lactose fermenting organisms. Okay? Also, selective din siya. Okay, so Macaulay is also selective. Okay, so it contains peptone, meat extract, sodium chloride, bile salts, lactose, and a neutral red indicator. Okay, kaya siya naging selective kasi particularly used lang siya for for enteric bacilli. Okay, so ayan, as for example, Escherichia, Enterobacter, Proteus, Salmonella, mga bacteria found in your digestive system. Okay? And also, it contains bile salts. Okay? So, bile salts causes the medium to be acidic. Diba, normally, ang ating most bacteria, they survive in a medium na neutral si pH, around 7. Diba? So, kapag acidic, certain bacteria lang yung kayang tumubo sa kanya. Okay? We also have what we call sugar media. So, for the identification of most of the organisms, sugar fermentation reactions are carried out. For example, yung kanina, si Macaulay agar, we use lactose. Okay, so we differentiate bacteria, yung mga lactose fermenting, at yung mga non-lactose fermenting. So, other medias contain different types of sugar. For example, glucose, sucrose, mannitol, yan. So, carbohydrate fermentation is used for characterization and identification of bacteria, particularly important in the study of gram-negative bacilli. So, the usual sugar media consists of 1% of the sugar in peptone water along with an appropriate indicator. So, a small tube is kept inverted in the sugar tube to detect gas production. Okay, so, ayan. So, sorry. So, ito yung tube. Tapos, meron pang tube na naka-invert nasa loob. Okay? Okay, so ang tawag, sorry. So, sorry. So, ang tawag po dun sa tube na nakabaliktad is Durham's tube. Durham's, D-U-R-H-A-M. Okay. So, the medium is originally yellow. So, acid production is indicated by the development of pink color. So, acid production, nagpo-produce ng acid ang organism if it ferments, kapag fermenter siya, nung sugar na nandun sa solution or sa ating medium. Okay? So, an acid production is indicated by the development of pink color, as you can see with this one. And then, yung gas, makikita natin doon sa loob ng Durham tube. Compare mo dun sa negative, puro, ayan. And then dito nagkaroon siya ng space. So, it indicates na merong air sa loob. Okay, so meron siyang gas production. Okay. So, that is an example of a usual sugar media. Okay. Meron din naman tayong tinatawag na his serum sugars. Okay, these are used for organisms which are exacting in their growth requirements. Okay, like streptococci or pneumococci. And we have also transport medias. So these are holding medium designed to preserve the viability of microorganisms in the specimen but not allow multiplication. Okay? For example, yung sample natin is collected sa site A, pero sa site B pa siya ipaprocess. So you need a media para to make sure na pagdating ng site B, buhay pa rin ang iyong certain organism na gusto mong i-isolate. Okay, so yun yung purpose ng mga transport media. For example, COVID test, di ba meron tayong mga viral transport medium sa RT-PCR. So pag sinuwab si patient, yung swab natin, nilalagay natin sa transport media. Okay, so yung transport media, ang siyang nadali natin sa testing center. Okay? So, ganun. Ayan. Examples ng transport media, we have Amy's transport medium, and Stewart's transport medium, and Amy's are used for mga gonococci. Ito naman, buffered glycerol saline, usually used for enteric bacilli. Okay. Next, under reducing media is we have anaerobic media. So, anaerobic are used to grow anaerobic organisms. So, anaerobic ayaw nila ng oxygen. Okay? So, they contain reducing substances. Example natin, we have thioglycolate broth. Okay? This contain reducing agents such as yung sodium thioglycolate. The sodium thioglycolate, they act as a reducing agent to create an anaerobic environment deeper in the tube and allows anaerobic bacteria to grow. Okay, also contains glucose to maintain the anaerobic condition. It has vitamin C, cysteine, agar, to prevent convection currents of air, and methylene blue, which acts as an oxidation reduction potential indicator. Another example ng for anaerobic media is cooked meat broth or RCM broth. Robertson's cooked meat broth. So, yung itsura ng thioglycolate broth. Okay. So, let's focus first sa plated culture media preparation. So, ito yung gagawin ninyo on your activity. So, these are the materials that you're gonna be needing sa inyong sa ating activity. So, we will need the agar powder, so yung blood agar base. Distilled water, magdala po kayo. Alam ko meron pa ata kayo sa school. Petri dish, sterile petri dish. Mga Erlenmeyer flask, cylinders, pipettes, hot plates, stirring rod, spatula, analytical balance. Yun, meron tayo dun sa school. pH meter is, wala tayo nito pero wag na kayo magdala. Aluminum foil, pinagdala ko na kayo last time, so meron kayo. Alcohol lamp, weighing boat, this is paper lang. Ziploc, if possible, magdala kayo ng Ziploc, yung malaki, as in, malaki talaga ako. Ano yung pinakamalaki na kaya nyong mahanap? Lalagyan ng mga petri dishes. Parafilm, autoclave, parafilm, ata, parang hindi na siguro. Kasi meron namang aluminum foil. Autoclave, incubator, ref, water bath, autoclave, plug. Okay, so meron yun. So, yun lang dadalhin nyo. Distilled water, yung mga inyong petri dish, agar, ziploc. Aluminum foil, pasamahan na rin ako ng bulak. Yung malalaki po, yung nakaroll. Okay? Pero hindi naman ganun karami ha. Kahit yung pinakamaliit lang na nakaroll na bulak, hindi siya cotton balls. So for us to make, to prepare a agar, so ito yung sinusunod natin na computation. Okay? For us to know how much water and how much agar powder ang kakailanganin natin. So, our formula is, ito, W1 times V2 equals W2 times V1. Okay? So, W1 is the known formula weight of media. And W2 is the needed weight of media powder. So, known formula weight of median, makikita po natin yan sa packaging ng ating, packaging nung agar ninyo. Okay? And then, needed weight, yung ating X. So, yun yung hahanapin natin. So, volume 1 is the known formula volume of media. Yung makikita na din natin sa inyong packaging. For example, for 1 liter of media, 25 grams ng agar powder. Okay? So, yung constant yun nakikita sa packaging, sa instructions. Okay? And then, volume 2, yung desired amount of media. So, paano mo malalaman yung desired amount mo? So, computein mo, ilang plates ba ang kakailanganin mo? Okay? So, ang standard plate natin is around 25 ml. So, sa isang plate, meron siyang 25 ml of media. Okay, so 25 times the number of media needed, kunyari 40 plates yung kailangan mo, so 40 times 25. So, yun yung magiging desired amount mo. Okay? Plus 200 ml pa. Okay? So, yung makuha mo dito sa amount of volume plus 200 ml, yung 200 ml is meant for in case na may matapon, and in cases of spills or miscalculations. Kaya may extra tayong pa-extra na 200 ml. Okay? So, yun yung ilalagay mo sa desired amount of media. Okay. So, this is an example of sample computation on making or preparing medias. Okay, so, read the following questions based on your understanding of how to compute the media based on the instruction provided by the manufacturer. Okay? So, for example, ang situation natin is, ang nakalagay sa ating directions is to spend 28 grams of nutrient agar powder in 1,000 ml of distilled water. Okay? So, then dissolve by heating until boiling. So, autoclave for 20 minutes, then let it cool and pour in petri dishes. So, if we need to prepare 1,000 ml or 1 liter of nutrient agar medium, so we should dissolve 28 grams of media powder down. Okay, so the amount of liquid media poured in each petri dish is about 25 ml. Okay. So, with the given info, so try to answer po yung following problems. Okay? So, this one, balikan natin to sa face-to-face natin ha. Kasi, ang hirap magsulat dito using the mouse. Okay? So, para mas maintindihan po natin. So, for now, parang little sit work muna for you guys. Okay? Okay. So, the first step of our procedure is calculation and weighing. Okay. So, measure your computed volume of distilled water in a 1 liter graduated cylinder. Then place the weigh boat. Yung weigh boat is something that you can use lang na lalagyan ng powder. Okay, in our case, gamit lang tayo na paper boat para lalagyanan lang. Okay? So, place the weigh boat in the analytical balancer and weigh or press stir to zero out the weigh boat. Use a spatula to weigh your computed agar powder, okay, to the weigh boat. So, do not return excess material that is weighed out to the container kasi contaminated na yun, hindi na yun sterile. Okay? So, and then add the water to the flask. Next is to mix the water and the agar powder through boiling. Okay, so i-heat natin siya sa hot plate. So, prepare the hot plate or the water bath. So, after the powder has been dissolved, place the flask to the hot plate and then heat for approximately 10 minutes. So, add the remaining water in the graduated cylinder and continue to stir using the stirring rod. So, when you are sure that all the powder has been dissolved, turn off the heat or remove the flask from the water bath. Kung nakakita na kayo ng luluto ng gelatin, ganun lang din siya. Okay, so continuous lang ang stirring, tapos babantayan na hindi siya mag-boil over sa flask. Kasi minsan kapag bigla siyang kumukulo, tumatapon siya sa flask. So, yun yung babantayan natin. So, habang hinahalo, monitor the temperature, tingnan kung kukulo na siya. And then kapag yan naaamba na, ialis agad sa heat kapag aapaw siya sa flask. Okay? Di ba ganoon naman magluto ng gelatin? So, 10, use a pH meter or a pH tape to see if the media requires pH adjustments. Please. So, if in case na kailangan, to adjust the pH, 0.1 to 1 normal hydrochloric acid or NaOH can be used. Next, cover the flask using a cotton plug, okay, and secure the cover using foil. So, make sure it is fully covered. So, label the flask by taping the mouth of the flask and then set aside. After that, sterilization na tayo. Autoclave, i-autoclave natin yung inyong ginawa. Okay? So, add distilled water inside the autoclave compartment up to the level indicator and then place the flask with the mixture into the autoclave basket. Close the lid and then start na. So, 1 to 1 degrees Celsius at 15 minutes. 1 to 1 degrees Celsius at 15 psi for 15 minutes. Sorry. Next, after that, okay, so while the media or so in autoclave natin, while it is on the autoclave, arrange the Petri dishes plates on the counter. So after the cycle ends, check if the pressure is 0 first before Mo opens the autoclave. Mix the agar gently. Remove the basket using autoclave glove. Mix the agar gently while it is closed and then set the flask aside and allow it to cool for about 45 degrees Celsius. Okay, so before transferring or pouring into the Petri dishes. So for BAP preparation, nagagawa tayo ng blood agar plate. So allow the media to cool before adding the sheep's blood. Okay, so sheep's blood ang ginagamit natin for blood agar plates. For cup preparation naman, add the blood when it's still hot. Again, ang difference lang nila si BAP at si cup, yung cup is himalays na si blood. Okay, kaya mainit pa lang inilalagay na natin para maluto yung dugo. Okay, dispensing. Yan. So prepare the Petri dishes, making sure they are clean. Label each plate with the name of the media, date of media preparation, and your group number. Place them right side up in front of you. The bottom of the Petri plate is smaller and deeper than the top, which is the lid. So therefore, right side up is when the bottom is intact with the bench and the larger lid covers the bottom. Pour the agar into the Petri dish by lifting the lid slightly open to prevent airborne contamination. So ideally, this is done inside the biosafety cabinet or between two Bunsen burners or alcohol lamps to avoid contamination. So since hindi naga ng ang biosafety cabinet natin, sa desk lang tayo or tabletop magpe-perform. So repeat the procedure for the remaining Petri dishes. So you must work quickly now to prevent the agar from solidifying before you are finished pouring the plates. So note the volume does not have to be exact. However, make sure to cover the entire bottom surface, which is about 25 ml for standard size Petri dishes. And then follow aseptic technique. Next is packing and storage. Okay, so yun yung purpose ng Ziploc. Okay. So once the agar is dry, malamig na. Okay. Yan. Set the plates at room temperature and wait for them to cool. And then, seal the plates by using parafilm. Once the agar solidifies, place the agar upside down in Ziploc bags. Okay, seal and label the bag with its corresponding contents and the date of preparation. Store the agar in the refrigerator to prevent the agar from drying out and prolong its shelf life. Instead of parafilm, if you don't have any, I think we could use kahit tape or micropore. Pang ano lang naman, para lang masil. And that's it. Okay? So, yun. So, please be prepared na lang po sa ating laboratory once face-to-face classes have resumed. Okay? So, thank you po for listening. And if you have any questions, feel free to message me na lang. Okay? Thank you.